Good afternoon, students, and welcome to practice problem set week 6. We are slowly moving towards advancement of this course. It's already 3 p.m., but we will wait for 5 more minutes so that more participants can join, and then at 3:05 we can start the presentation. Yeah, it's 3:05 already. Let me share my screen and start this session. So I have written something in the chat box. It's the same set of instructions that we used to follow from the practice problem session 1. So all the participants, please turn off your mics and your video chat. You can communicate with me through the chat box. So this session will be an interactive session, of course, and we'll be trying to solve 10 practice questions corresponding to lecture week 6. And then we will move forward with each question after knowing the correct answer and the explanation. And I encourage all the participants to try answering these questions. With that much of an instruction, I'll share my screen. Yes. So meanwhile, I'll keep admitting students who are requesting to join this call, and let's start without any further delay. I hope you can hear me clearly and see my slides, see them changing. If yes, then you can just let me know in the chat box. Can you hear me properly and see my slides? Can you see them changing? Just let me know in the chat box and we will start. Yes, thank you. Cool. So I welcome you everyone again to NPTEL Nanotechnology in Agriculture. And what we'll be dealing with today is practice assignment for week 6. We had a set of lectures for week 6. Currently, the last series of lecture for week 8 is live on NPTEL site. You can go and follow your lecture series. And if you haven't caught up with the earlier lectures, I would advise you to do the same. The course instructor for this course, Nanotechnology in Agriculture, is Professor Mainak Das, and my name is Ashutosh Behra. I'm a PhD PMRF scholar from IIT Kanpur, and I'll be helping you solve these practice problems. Right? Let's move on to the first problem. So, it's the same strategy we will follow. We'll take up each question, and I'll give the question to you in my presentation and audio format. You can look at the question and try to answer in the chat box which option do you think is the correct answer. I'll wait for some time, collect some answers, and then let you know what is the right answer. Then slowly we will move on to the rationale behind what is the right answer and the explanation behind it. And moving like this, we'll complete 10 such questions in a span of around 2 hours. We'll try to complete. And if you have any doubts regarding these problems and practice sessions or the lecture week 6, you can ask post these problem solving session. If we have time, I can take a few of them. Okay. So moving on with the first question. First question says, which of these is not a nutraceutical? This is the first question. Which of these is not a nutraceutical? Option A, protein powder. Option B, aspirin. Option C, ashwagandha. Option D, lutein. What do you think, students, is the right answer? You can let me know in the chat box which of these options do you think is the right answer. Which of these is not a nutraceutical? Protein powder, aspirin, ashwagandha, or lutein? I'll wait for a minute for to collect some answers in the chat box, and I encourage all the participants to just try out the question, recall something, some information related to whatever you have learned in the course, and try to put it on the comment box. It's okay if your answer is wrong. We will move ahead and see what is the correct answer. I'll wait for a minute for students to answer. I hope you can see the question in my slides, and I'm reading it aloud also. Which of these is not a nutraceutical? Protein powder, aspirin, ashwagandha, or lutein? Okay, there are some answers. Soumyadeep is saying B, aspirin. Nirav says A, protein powder. Roshan is saying B. Okay, seems like we have less participants today. only 16 people. Fine, if students, it's up to the students whether to join the class or not. Let's move on with the answer. The correct answer to this question is option B, aspirin. Aspirin is not a nutraceutical. So Roshan and Soumyadeep are correct. Aspirin is the answer. So let us understand first what is a nutraceutical. A nutraceutical or a bioceutical is a pharmaceutical alternative which claims physiological benefits. So nutraceutical, if you break down the terminology, nutra means nutrition, something which is providing nutritional supplement or nutritional physiological benefits, as we see, which is similar to that kind of a pharmaceutical. In pharmaceutical, we take a medicine or a drug or a molecule which will help us fight infections, diseases, or better the betterment of the existing physiological condition. So nutraceuticals can be dietary supplements, like we take vitamins and coenzymes, carnitine. These are all dietary supplements. Micronutrients, vitamins, minerals, again. Functional foods. There can be probiotics. You must have heard about Yakult yogurt, probiotic drink, and also those are also nutraceuticals, providing you with a good amount of lactobacillus and other friendly bacteria which will help enhance the function of the body. Terpenoids, phytosterols, these are all again some of the nutraceuticals. Polyphenols, anthocyanins, flavones. So polyphenols, these anthocyanins, flavones are antioxidants which will help you fight the harmful reactive oxygen species. Glucosinolates, garlic, onion. So these are all part of nutraceuticals. Nutraceuticals are substances that are considered as foods, parts of foods, or dietary supplements that may claim to provide medical or health benefits fall under this category. So nutraceuticals are basically food or parts of food or dietary supplements. So if you look at all these options, protein powder is a nutraceutical because protein powder is giving you some supplement. It's giving you a supplementary intake of protein. Ashwagandha is an Ayurvedic, I think, Ayurvedic root of a plant or something, which will again give you nutritional value. Lutein is also a kind of uh, nutraceutical, but aspirin is a pharmaceutical. That's what makes it different. So examples, some of the examples of nutraceuticals include fortified dairy products, example milk, and citrus fruit like orange juice and all. So mostly food materials which provide nutrition fall under the category of nutraceutical. While a pharmaceutical is a substance used in the diagnosis, treatment, prevention of disease, and for restoring, correcting, or modifying organic functions. So it is likewise, when I spoke of what a pharmaceutical will help us fight or prevent a disease, restoring normal function. It will not enhance the nutritional value of the body or physically supplement any nutrition, but it will help us fight disease and prevent infection. They have contributed to improvements in life expectancy and quality of life of many patients. So, invention of discovery of pharmaceuticals lengthened the life expectancy of many patients because patients which earlier used to die because of infections and some diseases, pharmaceutical extended the life of these patients. And aspirin is a very commonly used pharmaceutical. It is used as a painkiller, an analgesic, antipyretic, and anti-inflammatory. So antipyretic means to cure fever, anti-inflammatory to suppress the inflammatory signals, analgesic as a painkiller. So aspirin is the correct answer. Aspirin is a pharmaceutical but not a nutraceutical. While protein powder, ashwagandha, and everything else, lutein, are nutraceuticals. Is it clear to the students? I hope you understood the explanation. You can just let me know in the chat. We'll move on to the next question. Yes, yes, nice, cool. So the second question, which of the following process is rapid cooling of liquid without ice crystal formation? So this is a definitive question. This was taken up in the lecture week 6. If you try to remember, a process of rapid cooling of a liquid without ice formation. Option A, crystallization. Option B, cryogenics. Option C, solubilization. Option D, vitrification. So this is a very important, you may say, tricky technique. It is used mostly in preservation. I gave you almost many hints. So this technique is used in preservation. Now you just have to recall where was it said in the lecture, or if you have any knowledge about it. I'll wait for a minute and collect some answers. There are already many answers. Wow. Soumyadeep, Roshan, Priyanka, Neerav, everybody is saying de-vitrification. Currently, there are 21 participants. Okay, today's attendance seems quite thin. No issues. Let me wait to collect some more answers. So, students, please try to answer. It will benefit you as well as me to carry forward this discussion. It's okay if you're just guessing or if your answer is wrong. Do not shy out, be confident. Which of these option do you think is the correct answer to this question? Rapid cooling of liquid without ice crystal formation. There are already some answers. People are majorly saying D. I want other students to answer. Wait for a few seconds and then we'll solve. Okay, we'll move forward with this question. So the correct answer is D, vitrification, which was correctly answered by all these students. Congratulations. Soumyadeep, Roshan, Priyanka, Neerav. So let us understand one by one what are all these processes. The first option is crystallization. Crystallization is a natural process which happens when materials solidify from a liquid or as a precipitate out of a liquid or a gas. So crystallization is a process in which solid materials precipitate out or solidify from a different phase, such as liquid or a gaseous phase. So anything that could be called crystallization. And they form certain organized structures called crystals. They form lattices. That is the process of crystallization. Crystals can be made out of one species of atom, different species of ions, or even huge molecules like proteins. So it's not always that only materials can form crystals. Even atomic species can form crystals. Ionic species like NaCl, you have sugar cubes. We have sugar cubes. Sugar is made up of sucrose. We have NaCl. NaCl is an ionic compound. NaCl, we have salt crystals. So crystals can be of variety, many varieties, and proteins. So crystallization property of proteins is exploited in a technique called X-ray crystallography, in which protein crystals are set, and the structure of protein is determined with the help of X-ray crystallography by passing X-rays into the protein crystal and seeing and measuring the deflection of the X-rays to form a diffraction pattern, right? So crystallization can be of many materials, and this is how crystals look like on a petri dish. If you provide the right conditions, then many materials can precipitate out from liquid or gas phase to form crystals. So many minerals in the underground ore formation metals occur in crystals. What is cryogenics? On the other hand, cryogenics is the science that addresses production of very low temperatures. So cryogenics will deal with very low temperatures. So lowering the ambient temperature of a substance or in an environment effectively is the science of cryogenics. Cryogenics is used in a variety of applications. It can be used to produce cryogenic fields for rocket, MRI machines that use liquid helium and require cryogenic cooling, storing large quantities of food, special effects fog, freezing blood and tissue samples, and even cooling superconductors. So cryogenic science, mostly we use liquid nitrogen in our lab or in uh, day-to-day cryogenics facility, because liquid nitrogen can produce very low temperatures or sub-zero temperatures, and then this cryogenics can be used in rocket fuel rocket fuel, because burning of rocket fuel produces a lot of heat energy. To cool down that, we use cryogenics. For freezing blood and tissue samples, for preservation of tissues and blood is an intact, for storing food, and for cooling of superconductors. So everything, many fields there are which use cryogenics. And here is an image showing the use of liquid nitrogen as a cryogenic media, which will freeze the tissue sample. So generally you must have heard about bacterial stock preservation or cell culture stock preservation. So when you have a immortalized cell line or a bacterial stock, we provide a cryogenic environment to the cells or the bacteria in the form of glycerol for bacteria or DMSO for the mammalian cell culture, and then we cryo freeze it in a jar of liquid nitrogen, which will instantly freeze them and drop them at the current state in which they are present in the DMSO or in the glycerol. So in that way, we are preserving the cell line or the bacterial line for future use. So whenever we want to use that cell line further in the timeline, we revive that cell or we reculture that bacteria. So we preserve the viability of many living things, such as freezing blood, tissue samples, or food. All can be achieved through cryogenics. So that is cryogenics. We learned about crystallization, cryogenics. Now what is solubilization? Solubilization is the increase in solubility of poorly water soluble substance with surface active agents. The solubilization is making a substance much more soluble in water. So if there is a hydrophobic substance, now a hydrophobic substance won't be easily soluble in water. So what we will do, we will add some surface active agents and increase the solubility of hydrophobic substance inside water. So that process is called solubilization. So it can be of any substance, even a hydrophilic substance which is already dissolving in water, but we want the hydrophilicity of that substance to increase. So we use some surface active agents. The mechanism involves entrapment of molecules in micelles and tendency of surfactants to form colloidal aggregations at critical micelle concentration levels. So this is an example of micelle formation in which there is colloidal aggregation of substances due to micelle formation, which increases the solubility of the substance in water. And here is the schematic showing an amphiphilic molecule, both lipid molecule having a hydrophobic tail and a hydrophilic head. There are few molecules which have lesser dissolution in water. Now when they are formed in a organized micelle structure with all their hydrophobic tails pointing inside of the structure and hydrophilic heads surfacing the water and plus oil droplets, so we can capture these oil droplets which are hydrophobic, which do not have high solubilization. Now we will add this micelle to this oil droplet, which will form which will cause the oil droplet to solubilize in water. So that is the process of increasing the solubilization of this oil droplet with the help of micelles. So this process is called solubilization of any substance. Here the substance is oil droplet. We are increasing the solubility of oil droplet in water. Now coming to the last technique or vitrification. Vitrification is an alternative approach to cryopreservation that enables hydrated living cells to be cooled to cryogenic temperatures in the absence of ice. So in cryogenics, we saw uh, use of sub-zero temperatures for freezing substances. But in vitrification, we can bypass the ice formation or crystal formation, ice crystal formation in a live sample or a substance that will enable hydrated living cells to be cooled to cryogenic temperatures. So how this process is done? We use something called cryoprotectants. Adding cryoprotectants to water prevents water molecule from gathering and forming ice. So these are some of the molecules like cryoprotectants which we talked about. We will add these molecules into the water, and now the water molecules cannot come closer and form ice. So instead of freezing, the molecules slow down their movement as they are cooled. So now the temperature will reach 0 degrees Celsius, but water will not form ice. And below some minus 100 degrees Celsius, molecules become more and more locked in place and a solid is formed. At around minus 130 degrees Celsius, the cells are considered vitrified. So vitrification can achieve temperatures as low as minus 130 degrees Celsius. So in that state, there is no ice formation still due to use of cryoprotectants, and all the living materials can be frozen, hydrated in the hydrated form and formed in a solid form to uh, preserve the vitality and functionality of the material. So this vitrification is used in seed preservation and germplasm preservation for propagating in agriculture and plant culture that was mentioned in lecture week 6. So we now learnt about all the four processes: crystallization, cryogenics, solubilization and vitrification. So the answer to our question was reaching of sub-zero temperatures or bypassing the ice stage, right? That was the question. Rapid cooling of a liquid without ice crystal formation, and the answer is vitrification, which all the students have answered very correctly. Is it clear to the students? Were you able to understand about all different process? You can let me know in the chat. Great, great, cool, nice. Let's move on to the third question. So this is a question about cerium oxide radical scavenging. During free radical scavenging of cerium oxide, cerium exists in which oxidation state or states? During radical scavenging of cerium oxide, the options are plus 3 only, plus 4 only, plus 3 and plus 4, or none of the above. So if you have some idea about the oxidation states or state of cerium in cerium oxide and how the cerium oxide can be used for radical scavenging of uh, different samples, you can answer this question. I'll wait for a minute and collect some answers. What do you think is the correct answer to this question? Mention in the chat box. Let me know in the chat box. What is the oxidation state in which cerium exists while radical scavenging? Oh, nice. There are already many answers. You guys are very fast. And people are answering option C, plus 3 and plus 4. Hmm. Great. Roshan, Soumyadeep, Neerav, Malarvili, Priyanka are all answering C. Okay. And the correct answer is also C, plus 3 and plus 4. You guys did it. Congratulations. You are reaching the correct answer. So cerium exists in plus 3 as well as plus 4 oxidation state in cerium oxide during radical scavenging. Free radical scavenging. Now let us learn what happens in a cerium oxide molecule when it is existing in a lattice. The ratio of C3 plus C4 plus in the CEO2 lattice, and thereby the concentration of oxygen vacancies is an important parameter for tuning the free radical scavenging activity of CENPs. So here CENPs means cerium nanoparticles. So what this statement tell us, the ratio of C3 plus to C4 plus atoms in present in a cerium oxide lattice, and thereby the concentration of oxygen vacancies because of presence of different kinds of atoms in the CO2 lattice, there will be oxygen vacancies in the lattice. Now these oxygen vacancies will help the CO2 lattice to scavenge different oxygen radicals or free radicals into that lattice to occupy that vacancy. So that is an important factor which can enhance the free radical scavenging activity of cerium oxide nanoparticles. C34C4 plus redox couple and the high mobility of non-stoichiometry induced oxygen vacancies in nano-sized CNPs are responsible for oxygen scavenging properties. So what do I mean by this complex terminology? It's just saying that C3 plus and C4 plus state are in continuous reversible redox reactions in which one state oxidation state, suppose C3 plus, forms another group of molecules with C4 plus, and this formation creates some oxygen vacancies. Let's say two molecules of C3 plus gets converted into two molecules of C4 plus. Now the total amount of the stoichiometry of C3 plus ions will be less by two molecules, and C4 plus will be more by two molecules. That is talking here about non-stoichiometry induced oxygen vacancies. Now when these conversions will happen due to redox couple reactions, this will induce non-stoichiometry induced oxygen vacancies, and those vacancies are responsible for oxygen scavenging properties of cerium oxide nanoparticles. So if you look at an MRI scan of a CEO2 lattice, so this is the structure of CO2, cerium oxide nanoparticle lattice. So there are many molecules. There are stoichiometrically balanced molecules of C4 plus and C3 plus. So all these pinkish materials are C4 plus here, brownish pink material, and the lighter color materials are C3 plus. And gadolinium is used as an MRI contrast agent. We encountered gadolinium in the last practice problem session, and this is showing here Ross scavenger activity. So there are many molecules. CEO2 does not comprise only of a single set of molecules. There are many molecules of cerium in 3 plus state as well as in 4 plus state. So both of these molecules exist, and their interconversion causes vacancy in the lattice, which is then acting as an oxygen scavenger for filling those vacancies in the lattice. And here is a schematic of summarizing the free radical scavenging and recovery pathway of C3 plus to C4 plus conversion. So if you look at here, Ce2O3, Ce2O3 is a cerium 3 oxide molecule where cerium is present in 3 plus stage, plus 2 OH minus plus free radical gives 2 CEO2 plus H2O. So this is the free radical scavenging. In this method, it will consume the free radical and produce two molecules of CEO2, which is cerium 4 oxide, where cerium is existing as Ce4. Now this Ce4 plus 2 CO2 can again give Ce2O3 molecules by removal of H plus ions or recovery. So which will recover the parent molecule Ce2O3, cerium 3 oxide, and this cycle will again continue. This cerium 3 oxide will replenish this pool, and again it can go for another round of free radical scavenging. So this transition from C3 plus to C4 plus and back again to C3 plus, this occurs frequently, which gives the cerium oxide nanoparticle its oxygen scavenging property. So this is a schematic showing the radical scavenging of cerium oxide. So the answer to our question would be cerium exists in both plus 3 and plus 4 states, while uh, free radical scavenging in tissues or in wherever the application is there. I hope it is clear to the students. So we learnt about the reactions involving free radical scavenging of cerium, and which kind of molecules are present in the lattice structure of cerium oxide, and why does it act as an oxygen scavenger. Is it clear to the students? You can just let me know in the chat box. Yes, yes, got it. Yes, cool. It seems like there are very few students today, but it's okay. Maybe students are not able to join this class. Let's not bother about them and move on to the next question of this session. Question number 4. Which of the following is are commonly existing free radicals in living system? It's a question about free radicals again. Which of the following do you think are commonly existing free radicals in living systems? Option A, superoxides. Option B, hydroxyl. Option C, nitric oxide. Or option D, all of the above. Which of these do you think are commonly existing free radicals in living systems? Come on. I guess you know these answers since you have answered a cerium oxide nanoparticle very accurately. What do you think is the right answer? Please let me know in the chat box. I'll wait for a minute to collect some answers. Take your time reading the question on the presentation slide. What are the different kinds of free radicals present in the living systems? Okay, there are some answers I can see. Somyadeep, Roshan, Durga Devi, Joshi are all saying D. Neerav was saying A, but he changed to D. Superoxides, hydroxyl, nitric oxide, or all of the above. Oh, there is a comment. Sir, all radicals are present. And the answer is option D, all of the above. Very good. Very good. Congratulations to all the students. You are right. All these are free radicals which are present in the living system. Not just superoxides. Superoxides are a major group of radicals representing the reactive oxygen species, but there are also hydroxyl groups and nitric oxide which are also free radicals generated in the living system, right? So the answer would be option D, all of the above. Now, what are free radicals? Free radicals are oxygen containing molecules in our context because we are studying oxygen free radicals. Free radicals are oxygen containing molecules with an uneven number of electrons. So we know oxygen has 8 electrons, right? Oxygen has 8 electrons, but suppose one of the electron is missing from the outer shell. So in an oxygen atom, if you look at, the inner shell contains 2 electrons and the valence shell contains 6 electrons. So if one of the electrons is missing, now we have 2 and 5 configuration. That will be a free radical of oxygen. Or we have an extra electron. Let's say we have 2 and 7 configuration. We have 9, total of 9 electrons. Then that will also be a free radical of oxygen. So if we have uneven number of electrons and that atom exists in itself without in conjugation with any other substance or element, then that is called a free radical. And what does this uneven number of electrons do? They allow them easily to react with other molecules. Because of these uneven electrons, the oxygen molecule now is very reactive. It can either the accept one electron to fill its vacancy, or give that extra electron to somebody or to an element to uh, to suffice its valency. That's why oxygen with uneven number of electrons is very reactive. For that reason, they are also known as reactive oxygen species because they can react readily with other molecules, other stable molecules. And these free radicals can cause large chain chemical reactions in our body because they react so easily with other molecules. So in our body, leave about our body. In a single cell, if you see, there are many complex compounds and molecules and chemistry involved in maintaining a homeostasis of a single cell. So if you just look at the lipid bilayer of a cell, lipid bilayer is made up of lipid molecules, and lipid molecules are amphiphilic molecules with a hydrophobic tail and a hydrophilic head. So all these contains carbon chain backbone, carbon, hydrogen, nitrogen, oxygen, and these are all stable compounds. So if there is a free radical containing oxygen, it can readily donate its electron or fill an electron from the lipid backbone or a lipid entity and cause the disruption of a plasma membrane because it can readily react with any stable molecule. Similarly, these free radicals can react with proteins and cause them to misfold. So this is all the harmful oxidative effects of free radicals. That's why they can trigger large chain chemical reaction in our body because they can react so easily. And these reactions are called oxidation. They can be beneficial or harmful. So not all oxidation reactions are harmful. Some can also be beneficial, such as where we see during exercise and muscle contraction we need generation of reactive oxygen species in a subtle amount. So oxidation reactions can be beneficial or harmful. And here is a schematic showing the formation of free radicals and how it can affect our body. So in presence of UV light, oxygen present in the atmosphere can turn into hydroxyl radicals. Inflammation of the white blood cells. Mitochondria is a major source of reactive oxygen species in our cells. Smoking can also induce uh, production of free radicals. Ionizing radiation or air pollution, which also has effect from UV, and all these reactive oxygen species can go inside the cell, go inside the nucleus, and damage the DNA of a cell of an organism. So reactive oxygen species production is also also the hallmark of cancer. So all these DNA mutations and everything which leads to cancer can be caused by reactive oxygen species because they are so reactive they can react with DNA bases and cause them to change the chemistry, and they can be generated by a many phenomena. So we that's why we take so many antioxidants in our diet to reduce the harmful effect of oxidative stress. Tomato and pomegranates are all very good antioxidants we take in our diet to reduce the harmful nature of free radical oxygen species. And these are some of the examples of reactive oxygen species and reactive nitrogen species. So all these are free radicals. When we talk about reactive oxygen species, we specifically talk about ROS, but nitrogen species can also act as free radical. Singlet oxygen, ozone, hydroperoxyl radical, hydroxyl peroxide, H2O2, hydrogen peroxide, OH radical, superoxide radical, lipid peroxyl peroxide, hypochlorous acid. All these are radical reactive oxygen species which can generate unevenly balanced oxygen atom, which can then be act very reactively. Similarly, we have examples of reactive nitrogen species. Nitrous oxide is a good example. Peroxynitrate, nitroxyl anion, nitrogen dioxide, dinitrogen dioxide, and nitrous acid, nitrile chloride. So all these are reactive nitrogen species which can also be generated in the living cells and can cause harm to the body in the form of oxidative damage due to the presence of unevenly balanced free radical ion. So these are the examples of some reactive species and free radicals, and we learnt about what are free radicals and why they are so reactive and what are the sources of their production. So the answer to this question would be superoxides, hydroxyls, as well as nitric oxide are all examples of free radical generated in the living system. Is it clear to the students? I hope you are getting what I'm trying to explain it to you. If yes, then please let me know in the chat box if you're clear with the concept. Great, great, nice. Let's move on to the next question. Question number 5. Excessive reactive oxygen species. So it is again in continuation with the reactive oxygen species questions. Excessive reactive oxygen species in the muscular system can lead to dashed. So it's specifically asking about presence of excessive reactive oxygen species ROS in the muscular system. Option A, contractile dysfunction. Option B, fatigue. Option C, muscle weakness. Option D, all of the above. Now, which of these disparity do you think will happen if there is an excessive production of ROS in the muscular system? Is it contractile dysfunction? Is it muscle fatigue, muscle weakness, or all of the above? Let me know what do you think is the correct answer in the chat box. For students who have not answered yet, I would encourage everybody to participate. Please participate. This is a two-way discussion, so that we uh, we at both ends can be beneficial. We at both ends can benefit from this. So please try to answer these questions. I'll wait for a minute and collect some answers. Which of these do you think is a disparity which can occur when there is an excessive production of reactive oxygen species in the muscular system? Okay, there are many answers. Roshan, Neerav, Somadeep, Joshi, Priyanka, Durga Devi, everybody is saying option D, all of the above. Great, great. And I see students are still trying to join the class. So let's see what is the correct answer. The correct answer is as mentioned by and as answered by everyone. It's D. So excessive reactive oxygen species can lead to all of these major disparities. Contractile dysfunction of the muscle, muscle fatigue, muscle weakness, everything. So all of these can happen due to excessive production of ROS in the muscular system. So what happens in a skeletal muscle system? How does ROS gets produced? Repetitive skeletal muscle contraction result in activation of mitochondrial biogenesis, fiber type transformation, and angiogenesis. So whenever a person is doing strenuous exercise, heavy loaded exercise, so there is repetitive skeletal muscle contraction and activity. So these repetitive actions will then cause mitochondria to be hyperactive, mitochondria biogenesis, and there will be angiogenesis. Angiogenesis is production of blood vessels. So much more blood vessels, muscle will require more blood for its repetitive contraction, and mitochondria will increase in activity. So together all these phenomena will increase the muscle's capacity of aerobic metabolism and its resistance to fatigue. So why does the body do that? Because now the muscle needs more energy. It needs more aerobic metabolism to generate some energy for that repetitive contractile motion. Now, high muscle activity also involves a strong increase in reactive oxygen species production. These unstable molecules and ion contain oxygen and are extremely reactive due to an unpaired electron. Now, what will happen due to higher activity of mitochondria, as we have learned that mitochondria is a source of ROS, reactive oxygen species, there will be increase in reactive oxygen species when a person does high strenuous exercise or repetitive skeletal muscle contraction. Now, these ROS molecules
are extremely reactive due to presence of unpaired electron, which we learned in the previous question. Due to presence of unpaired electron, it is very reactive, can react with stable molecules present inside the cell, which is harmful for the cell or the muscle. Among these oxygen intermediates are the free radicals, superoxide, peroxide, hydroxyl radicals, and other highly reactive oxidants such as singlet oxygen and hypochlorous acid. So all these examples we encountered, these are these are the most important free radical examples: superoxide, peroxide, hydroxyl radical, singlet oxygen. All these are very highly reactive and they can damage the normal homeostatic cellular system. They promote oxidation reactions with other molecules such as protein, lipids, DNA, and thus can highly be detrimental. So this is what I was speaking of. These singlet oxygen, superoxide, and other free radical oxygen species can react with such stable molecules like proteins, lipids, and DNA. It can damage the cell membrane by reacting with the lipids. It can cause mutations in the DNA. It can cause the proteins to lose certain amino acids or lose their three-dimensional structure or turn them inactive, so which can be detrimental, highly detrimental to the living cell. So this is a schematic explaining the relation of skeletal muscle contraction, energy, exercise, and ROS production. So during aging, physical inactivity and malnutrition. When we age, we have we have more physical inactivity, we have malnutrition. This can all lead to oxidative stress. So this is an example of aging related decrease in physical inactivity. So whenever there is high physical inactivity also, there can be higher production of reactive oxygen species. Now in aging individuals, oxidative stress can cause decrease in protein turnover because the proteins cannot be recycled in a faster way, reduced regenerative potential of skeletal muscle, impairment of muscle fiber activation at the neuromuscular junction. So neuromuscular junctions are areas where motor neurons release their synapses into the muscle cell. So these muscle fiber activation at the neuromuscular junction will be slowed down. Impairment of excitation contraction coupling the rear and cross and of cross bridging cycling. So these are all phenomena leading to muscle contraction and strength of the muscle due to cross bridging of different muscle cells. There can be increased strength, but which decreases with aging. As aging increases, oxidative stress increases, which leads to decrease in protein turnover, regenerative potential of skeletal muscle, and impairment of muscle fiber activation at neuromuscular junction. And all these activities lead in overall decline in muscle mass and strength, which is also known as age related sarcopenia. So this is a phenomenon which is triggered by oxidative stress or not enough oxygen due to physical inactivity, which generates deficient oxygen radicals or oxygen radicals with deficient electrons. Then they can all together go and lead to age related sarcopenia, which is decline in muscle mass and strength. So reactive oxygen species or oxidative stress are very harmful, and this is a schematic showing different disparity of contractile dysfunction, muscle weakness, and fatigue. So whenever there is ROS production, there is increase in blood pressure, increase in chance of atherosclerosis or blockage of arteries due to cholesterol deposition, decrease of antioxidants, the amount of antioxidants that we take when there is increase in production of ROS, it cancel out the concentration of antioxidant present, endothelial dysfunction in which endothelium or the single cell capillary linings, endothelium lining lines the arteries, veins, and other capillaries and the stomach lining. Due to nitric oxide NOS coupling with ROS, there is endothelial dysfunction. The arteries walls and the capillary wall will be thinning. Decrease of mitochondrial DNA will lead to mitochondrial dysfunction. Due to ROS, these ROS go and interact with mitochondrial DNA, cause mutations in them, and cause them degradation, which will lead to dysfunction of mitochondria and production of more ROS. cardiomyocytes hypertrophy, contractile dysfunction. So these are many of the phenomenons which are mediated through ROS. Via these are the signaling molecules like JNK, ERK1, 2, P38, MAPK, CAMK2. These are all proteins through which these signaling can occur and which can lead to contractile dysfunction, muscle weakness, fatigue, and everything. So ROS can cause all of the above. So the answer was it can cause excessive production of ROS, can cause contractile dysfunction, can cause muscle weakness and fatigue, and can also cause very many other health conditions like increase in blood pressure, atherosclerosis, and endothelial dysfunction. I hope it is clear to the students that ROS can cause many diseases due to excessive production of ROS in the skeletal muscle system. Is it clear to the students? You can just let me know in the chat box. Great, great, nice. Let's move on to the sixth question. Now, this is a memory based question. Which of the following is vitamin E? I hope everybody should answer this very correctly. Which of the following is vitamin E? Option A, retinol. Option B, ascorbic acid. Option C, tocopherol. Option D, cerium oxide. I mean, you must have heard about different vitamins and their names, their properties, which foods are rich in them. I hope you can just recollect them and let me know what is the correct answer. What is vitamin E otherwise known as? Is it retinol, ascorbic acid, tocopherol, or cesium oxide? You can answer in the chat box. I'll wait for a minute to collect some answer. I can see some 4 5 answers. Priyanka, Somadik, Durga Devi, Roshan, Joshi, everybody is answering C, tocopherol. As expected from you guys, tocopherol is the correct answer. Vitamin E is also known as tocopherol. Great, very nice, very nice. So let's see what is tocopherol and how does vitamin E look like. Tocopherol exists in 4 different forms designated as alpha, beta, delta, and gamma. So there are 4 types of tocopherol: alpha, beta, delta, gamma. They present strong antioxidant activities and it is determined as a major form of vitamin E. So the main function of tocopherol or vitamin E is to act as an antioxidant, is to protect our body from reactive oxygen species. And vitamin E major form is known as tocopherol. Tocopherol as a group is composed of soluble phenolic compounds that consist of a chromanol ring and a 16 carbon phytyl chain. So to simplify things further, this is a structure of tocopherol. So this is what the statement was saying. Soluble phenolic compound consists of a chromanol ring and a 16 carbon phytyl chain. So this is a chromanol ring. All means alcohol. So this is an alcohol with OH group here, chromanol. And this is the 16 carbon phytyl chain. This is the whole structure of vitamin E, tocopherol. And many foods such as avocados, spinach, bell peppers or capsicum, almonds, nuts, groundnuts, hazelnut, walnuts, raisins. So all these foods oils are rich in vitamin E, which can supplement us with the required dose of tocopherol. So that's why we are advised to take nuts and almonds in our daily doses. to increase the concentration of antioxidants for fighting the damage causing ROS molecules. And tocopherol is one of the major forms of vitamin E found in our body. Now, what is retinol? Retinol is also called vitamin A1, is a fat soluble vitamin in the vitamin A family found in foods and used as a dietary supplement. So, retinol is also the second name for vitamin A. It is a fat soluble vitamin. So, vitamin A, vitamin K are all fat soluble vitamin. Well, vitamin B, which is thiamine, riboflavin, so these are different vitamin B complexes, cyanocobalamin, vitamin B12. These are all water soluble vitamins. Retinol is also known as vitamin A, and as we know, retinol is very important for eyesight. Retinol or other forms of vitamin A are needed for vision, cellular development, maintenance of skin and mucous membrane, immune function, and reproductive development. So uh, we must have come across many statements saying that eating carrots will improve your eyesight. So all these carrots contain this vitamin A or retinol, which is therefore indirectly telling us that presence of retinol can increase our vision or better our eyesight. So this is an important compound, and these are the foods which are high in vitamin A: fish oil, beef liver, cheese, spinach, bell pepper, butter, green onions, pumpkins, eggs, broccoli. So these are all carrot is an important source of vitamin A or retinol, which can be used for vision, immune function, mucus, and reproductive development. Now, what is ascorbic acid? Vitamin C is also known as ascorbic acid or ascorbate. Is a water-soluble vitamin found in citrus and other fruits and vegetables, also sold as dietary supplements. All the citrus fruits that we eat, lemon, beet, lemon, oranges, all these contain vitamin C or ascorbic acid, the sour form. All the sour foods contain vitamin C. Ascorbic acid is essential nutrient involved in repair of tissue, formation of collagen, and enzymatic production of certain neurotransmitters. So vitamin C is necessary for production of neurotransmitters, which is an important aspect of nervous system. So and vitamin C deficiencies causes scurvy. Scurvy is the damage of gums, which is due to deformation of collagen. So vitamin C or ascorbic acid is needed for active formation of collagen, which makes our gums and other extracellular matrices. And these are certain foods as we spoke of: oranges, grapefruit, kiwi, sweet pepper, papaya, lemon, strawberry. So all these fruits contain vitamin C because they have this sour taste. Any fruit containing vitamin C will have a sour taste. And we take vitamin C in day-to-day life via lemon or oranges, which will keep our gums and teeth very fine due to production of collagen. So in this explanation, we learnt about tocopherol, retinol, and ascorbic acid. So vitamin E is also known as tocopherol, but not retinol and ascorbic acid. Clear to the students? Is it understandable? Did you get it? Let me know in the chat if it's clear to you. Great. Nice. Let's move on to the next question. Question 7. Which of the following was noticed in exercising mice with doses of cerium oxide nanoparticles? So this is a question based on experimental analysis or from a research paper where they were exercising mice or hamster on a hamster wheel with doses of cerium oxide nanoparticles. So what were the phenomenon that was observed? What was the data that you think could be observed? Option A: increase in glycogen in the mice. Option B, increase in muscle fatigue. Option C, reduction in ATP content. Or option D, fewer motor neurons. This could be a difficult question. So I'd give you guys ample time to follow the question on the presentation. Which of the following was noticed in exercising mice with a dosage of cerium oxide nanoparticles? Option A, increase in glycogen. Option B, increased muscle fatigue. Option C, reduction in ATP content. And option D, fewer motor neurons. If I hint you guys, just have a thought about what could be the role of cerium oxide nanoparticles. What is the function of cerium oxide nanoparticles? And then you will be able to reach this answer. Now, if a muscle or a living tissue is treated with serum oxide nanoparticles or mice exercising with serum oxide nanoparticles, what could be the phenomenon? Is it increase in glycogen, increased muscle fatigue, reduction in ATP content, or fewer motor neurons? I'll wait for a minute and let others answer. There are 4 5 students who are consistently answering. Very good, keep it up. But I want other students as well to participate. Which of these do you think will happen if you provide a living tissue with a dosage of cerium oxide nanoparticles? I'll just wait for a minute and then collect some answers. Yeah, seems like people are not confident enough. It's okay. You can try answering these questions even if you are not sure of the correct answer. You can just guess what is the correct option. Let's move on with the correct answer. Wow, it's option A. So all of you guys are answering very correctly, accurately this time. Very good. So there will be an increase in glycogen if a mouse tissue or in exercising mice, muscle tissue is treated with a dosage of cerium oxide nanoparticles. What will be observed is an increase in glycogen. So what happens in a skeletal muscle system? Exercising mouse here means the mouse is running on a hamster wheel. It's some kind of an exercise, physical skeletal muscle activity for the mouse. Small amount of ROS generated during mild exercise is essential for normal force generation. See, a small amount of ROS is required for normal force generation, whereas large quantity of ROS generated during intense exercise may cause contractile dysfunction resulting in muscle weakness and fatigue. So this is what we studied in 2 questions prior, that if there is an increased ROS production, then due to intense exercise, that can cause contractile dysfunction in the muscles, leading to muscle weakness and fatigue. But a small amount of ROS is required for normal function. Now, what happens when you treat the muscle with cerium oxide or nanocerium oxide nanoparticles? Nanocerium oxide coexists in two oxidation states. This we have thoroughly encountered in the previous questions. C3 plus and C4 plus are the two different oxidation states in which nanocerium oxide exists when treated into the mice muscle and undergoes an autocatalytic swapping of the oxidation states by virtue of its unique surface properties. So as we as we saw in the earlier cerium oxide nanoparticle functioning, The lattice will contain C3 plus as well as C4 plus molecules, and there will be an autocatalytic swapping of the oxidation states, which will create vacancies where the extra electron from oxygen or the free radical can come in. That's how the nanoserium oxide will function as an oxygen scavenger. The pharmacological activities of CON or nanoserium oxide have been attributed to the multi-enzyme mimetic activities such as superoxide dismutase, catalase, and peroxidase. So what this statement says that treatment of cerium oxide nanoparticles or nanoceria to the muscle can mimic the activity of superoxide dismutase, catalase, or peroxidase. Superoxide dismutase will scavenge superoxide radicals. Similarly, catalase and peroxidase will catalyze other scavenging of other radicals such as peroxide radicals and singlet oxygen radicals. So treatment of nanoceria can give the same kind of effect mimicking the enzymes SOD, catalase and peroxidase. Further, nanoceria or CONs represents for nanoceria reportedly quench cellular reactive nitrogen species also. So apart from not only uh, doing scavenging the reactive oxygen species, nanoceria or cerium oxide nanoparticles can also scavenge reactive nitrogen species from the cells, degrade strong oxidizing agent. So nanoceria can quench the cellular reactive nitrogen species, RNS reactive nitrogen species, degrade the strong oxidizing agent peroxynitrite, and promote survival during oxidative stress in an animal models. So it has experimentally been verified that nanoceria can quench the reactive nitrogen species and degrade strong oxidizing agent peroxynitrate, and promote survival. And again, this is the reaction mechanism which we encountered in the previous slides also. So lattices with C3 plus atoms can scavenge superoxide radical C3 plus O2 superoxide plus 2H plus to give C4 plus H2O2, which will lead, and Ross here molecules are H2O2 and O2, and then C4 plus scavenging Ross while regenerating surface. This can also scavenge reactive oxygen species and swap into C3 plus oxidation state. C4 plus H2O2 gives C3 plus H plus HO2. So following this reaction, the C4 plus molecules can then back converted into C3 plus molecules, and this cycle can continue while they can scavenge the reactive oxygen species as well as reactive nitrogen species. Now in this research paper from Aditya Arya et al. 2017, which is from the lab of Professor Mainak Das, they did this experiment in which they injected the mice with cerium oxide nanoparticles or ceria in certain concentration. So here in the panel B, if you see control mice, so this is a view of muscle of the mice. Control, which is untreated with any particles, is the wild type. Here it is treated with cerium nanoparticles. This control mouse is made to exercise, and CNP mouse is also made to exercise. So difference between the muscle mass, intact muscle in situ, isolated gastrocnemius muscle. So this is showing the muscle mass isolated from the wild types, ceria nanoparticle, exercise, and CNP exercise. So if you see, CNP plus exercise is leading to a greater muscle mass, because as we know, if you do more exercise, there will be increase in muscle mass, but doing more physical activity or exercise which also generate ROS, which can degrade the muscle mass. Due to presence of cerium nanoparticles, now this detrimental effect will be removed, so there will be an happy increase of muscle mass. So if you look at panel A, the blue bars represent control mice, red bars represents ceria nanoparticle treated mice. Average running time of the mice here shows that in exercise sedentary mice or static mice, ceria oxide nanoparticles mice average running time is more, and in exercisable mice this is more significant. It states that ceria nanoparticle treated mice can run for more times in exercise mode, because now the reactive oxygen species that is produced by the exercisable muscle is captured by the ceria nanoparticles. Similarly, we have other panels here showing wet weight of the muscle mass, which shows that ceria treated nanoparticle muscle mass is more than control mice muscle mass. And this is the panel of interest to us, which also shows that there is an increase in glycogen concentration in ceria nanoparticle treated muscle of the mice when the mice are exercising. So there is an increase in glycogen content, and this here shows the levels of lactate content. So lactate is a lactase lactate metabolism is a sign of fermentation, lactic acid fermentation pathway, in which the end product of glycolysis pyruvate is converted into lactate or anaerobic respiration, which is detrimental to the muscle, which provides immediate spot of energy to the muscle but might be detrimental due to accumulation of lactates in the muscle. So you can see clearly that in ceria nanoparticle treated muscle, in exercise module, the levels of lactate in the mice muscle is lesser, showing that it is less toxic. So application of ceria nanoparticles to mice muscle showed improved results, increase in glycogen, increase in running time, increase in muscle mass, and decrease in lactate levels, harmful lactate levels. So in summary, what the answer that we were looking for is glycogen levels increase when ceria nanoparticles are treated to mice and the mice are made to run. Is it clear to the students, the explanation? I hope you were able to understand how ceria nanoparticles are helping in these mice models, and what are the conclusions, different types of conclusion we can derive from this experiment and the modularity. I hope it is clear to the students. Please let me know in the chat box. Yes, great, great. Let's move on to the next question. Question number 8. Glycolysis is the pathway that converts glucose into dashed. It's a straightforward question. If you know anything about glycolysis, you might be able to answer this. It's an easy question. Glycolysis is the pathway that converts glucose to dashed. A. Fructose, B. Fats, C. Pyruvic acid, or D. Starch. I'll give you a minute, guys, to look at the question and write your answer in the chat box. You can also guess, do not shy out. We are almost reaching to last phases of this practice problem session. So all those students who have not yet answered and haven't gotten a chance to write down their answers in the chat box, please come forward and answer. I'll give you a minute and I'll wait to collect some answers. Which of these do you think involves glycolysis pathway where glucose is converted into dash? Okay, I can see many answers. Somadi, Priyanka, Roshan, Durga Devi, Nirav. I think they are all very prompt in answering. They have all answered C. And if you guys have little to brief idea about glycolysis, then you would know that answer C is the correct option. Pyruvic acid is the correct option. So the end product of glycolysis pathway is pyruvic acid. So in glycolysis pathway, we break down glucose. We use glucose as a starting material, break down the glucose into two molecules of pyruvic acid. A single molecule of glucose consists of 6 carbon atoms, and a single molecule of pyruvic acid consists of 3 carbon atoms. So we split a 6 carbon atom molecule into two molecules of 3 carbon atom molecules. In the process of glycolysis, starting material is glucose and the end product is pyruvic acid. And here is a schematic pathway giving you a brief idea about all steps of glycolysis. So I have just mentioned the important steps of glycolysis. So this is a 6 molecule glucose denoted by 6 rings here, and with the usage of ATP, 2 molecules of ATP, it forms an unstable phosphorylated form of 6 molecule of 6 carbon compound called fructose 1,6 bisphosphate. So doubly phosphorylated, the same 6 carbon molecule, but carbon atoms are rearranged now. Earlier it was glucose, now it is fructose 1,6 bisphosphate, and there are many enzymes involved in this process. Now generation of fructose 1,6 bisphosphate is the first important step, and this fructose 1,6 bisphosphate is unstable. So it breaks down into two 3 carbon molecule compounds called DHAP or dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. And these DHAP and glyceraldehyde 3 phosphate are interconversible. That means DHAP can convert into glyceraldehyde 3 phosphate and vice versa. So now we have broken down a 6 molecule carbon compound into phosphorylated form of 3 molecule carbon compound. And eventually all the DHAP will be converted into glyceraldehyde 3 phosphate. Now this marks the second step of glycolysis. Now for each molecule of glyceraldehyde 3 phosphate we will have the same set of reactions. That's why the whole process will occur multiplied into 2 times. So there will be reduction of NAD plus into NADH and generation of ATP in these steps, and finally to form a 3 molecule carbon compound called pyruvate or pyruvic acid. So this is the brief overview of how glucose, which is a 6 carbon compound, is first phosphorylated and turned into an unstable fructose 1,6 bisphosphate, which breaks down into 3 carbon phosphorylated compounds glyceraldehyde 3 phosphate, which then leads to production of energy while formation of 3 molecule pyruvate, and this happens in 2x times, so we get 2 molecules of 3 carbon compound pyruvate. So glycolysis is a metabolic pathway that converts glucose into pyruvate. The free energy released in this process is used to form the high energy molecules ATP and reduced nicotinamide adenine dinucleotide phosphate NADH. So ATP and NADH are all energy currency molecules. So in this process of glycolysis where glucose is broken down into pyruvate, ATP and NADH molecules are generated. Glycolysis is a sequence of 10 reactions catalyzed by different enzymes. So all these processes involve 10 different reactions catalyzed by 10 different enzymes. So if we summarize glycolysis, glycolysis takes place in the cytosol of a cell and it can be broken down into two main phases. So if you look at glycolysis, we can divide glycolysis into two main phases. One phase, the first phase is energy requiring phase, in which ATP molecules are used in the process of phosphorylating the 6 carbon glucose. In this phase, the starting molecule of glucose gets rearranged and two phosphate groups are attached to it. The phosphate groups make the modified sugar, now called fructose 1,6-bisphosphate, unstable, allowing it to split in half and form two phosphate bearing 3 carbon sugars because the phosphates used in these steps come from ATP. This is known as energy requiring phase because these phosphates come from the ATP molecule to phosphorylate the glucose molecule. Now in the second phase of glycolysis, which is energy releasing phase, what happens? Now that the 6 molecule carbon is broken down into 3 molecule glyceraldehyde 3 phosphate form. In this phase, each 3 carbon sugar is converted to another 3 carbon molecule through series of reactions, generating 2 molecules of ATP and 1 NADH molecule. Because this phase takes place twice, one for each of the 3 carbon sugars, it makes 4 ATP and 2 NADH overall. So in first phase of glycolysis, we have usage of ATP. 2 ATP is utilized to form glucose to fructose 1,6-bisphosphate, and the energy generation or energy releasing phase, we produce 4 ATP and 2 NADH. So net production of the glycolysis pathway is 2 ATP and 2 NADH. So this is a summary of how glycolysis takes place and what are the different important reactions taking place in glycolysis and how energy is generated and what are the different phases of glycolysis. And overall, glucose is broken down into 3 molecules. 6 molecule carbon, 6 carbon atom glucose is broken down into 3 carbon atom pyruvate at the end as the end product of glycolysis. Is it clear to the students? I hope I simplified down the glycolysis pathway, which will make you easier to understand. What are the important intermediate steps? What are the different phases of glycolysis? So that you do not have to remember all the named reactions. You just understand what are different concepts behind the phases of glycolysis and you will be able to memorize it. Is it clear to the students? You can let me know in the chat box. Okay, 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 great. Let's move on to the ninth question. It's again about cerium oxide. So this is a question based on chemistry. CeO2 converts Ce2O2 in what medium? In what medium will you expect CeO2 to convert into Ce2O3? Option A, basic medium. Option B, neutral medium. Option C, all mediums. Option D, acidic medium. In which medium do you think CeO2 will convert to Ce2O3? What do you think will be the right answer? Please mention in the chat box. I'll give you a subtle hint. If you calculate the oxidation numbers on cerium, you might be able to find out the answer. In which medium would CeO2 convert into Ce2O3? I'll wait for a minute to collect some answers. Come on, students, you can answer this. It's not a very difficult question. You just have to calculate what is the oxidation state of cerium in these compounds, and which medium do you think will cause the conversion of one oxidation state of cerium to the other oxidation state. I'm waiting for your answers in the chat box. It seems like students are having difficulty in answering this question. It's fine if you guess the answer and your answer is not correct. Just try to take a guess. What do you think will be the answer? I'll anyway let you know the correct answer and we'll understand. So see, if such questions come and you guys are not able to answer, it is a it is a sign to me that you are not able to understand these concepts. So I'll pay more attention to make sure that you understand all these concepts in the explanation part. So for that, I have to know whether you are able to understand this question or you are able to answer these questions. Then I'll pay much more stress and attention to the explanation part so that you do not commit this mistake again while you are uh, facing this kind of questions. Great, nice try. See, efforts are what we are looking for. Roshan is answering C. All mediums. Durga Devi, Nirav. Durga Devi is saying neutral medium. Nirav is saying acidic medium. So we have all different sets of answers. Let's see what is the correct answer. The correct answer is D, acidic medium. So I repeatedly gave you the hint to calculate the oxidation state of cerium in CO2 and Ce2O3. So okay, an additional question. Can you tell me what is the oxidation state of cerium in CO2 and Ce2O3? Can you mention in the chat box? I just want to see if you are able to calculate oxidation numbers uh, very effectively. What is the oxidation state of cerium in CEO2 and Ce2O3? Mention in the chat box. Then we'll move forward. What is the oxidation number on cerium in CO2? Let's say this compound CO2. What is the oxidation number of cerium? And in here Ce2O3, what is the oxidation number? What do you think? Come on, I think you will be able to solve this. We had taken many questions in the previous class describing what is oxidation number and how we can calculate the oxidation number of an element in a compound. Can you calculate the cerium oxide CEO2? What is the oxidation number on cerium? Let me know in the chat if you know what is the oxidation state of cerium in CEO2 and Ce2O3. Looks like students are struggling in these areas. Okay, let me not waste any more time. So see, I'll give you a trick how to calculate oxidation number of elements in a compound or in an ionic state or whatever. You take this compound, dissolve it in water, and see what is the different kind of ions this compound will produce. So if you dissolve CeO2 in water, so what is the valence of oxygen in oxygen? The valency of oxygen is 2 because it's short of 2 electrons. So when dissolved in water, oxygen will form a charge of 2 minus, right? So if you see oxygen will form a charge of 2 minus. Now there are two molecules of oxygen here. So 2 into 2 minus 4 minus. When oxygen, so a total charge on oxygen molecule in water will be 4 minus. Now to cancel out that 4 minus, cerium must be 4 plus, so that this compound can be neutral when it is uh, when it is in a compound state. So if you dissolve CeO2, if you dissolve cerium oxide in water, you will have cerium 4 plus and oxygen 2 minus, because oxygen valency is 2. So that will be the oxidation state. 4 plus 4 will be the oxidation state of Ce in CeO2. Similarly, in Cu2 Ce2O3, if you dissolve these oxidation states, if you dissolve this compound in water, what you will get is O2 minus again and C3 plus. These charges will exchange. So in this molecule we will have cerium 3 plus and O2 minus, while in this molecule we will have cerium 4 plus and O2 minus. So now the question arises, what medium will cause the conversion of C4 plus into C3 plus? So that is what will give the answer to our question. So if we see the explanation, so this is how cerium 4 oxide or CeO2 will look like dissolved in water. Ce4 plus and two molecules of O2 minus. So now the total molecule charge is 0. This is what I was talking about in calculating oxidation number of a compound. And this is how cerium 3 oxide will look like. Two molecules of cerium 3 oxide, cerium 3 plus, and three molecules of O2 minus, Ce2O3. Now these species are interconversible because of lattice properties of cerium. C3 plus state and C4 plus state are interconversible. C3 plus state to C4 plus state is called oxidation because it involves, it makes, it increases the oxidation number of C3 plus to C4 plus. So in a species, when there is increase in oxidation number, that process is called oxidation of the element. So C3 plus is changing into C4 plus. That means it is undergoing oxidation. And this process is called reduction, when C4 plus is decreasing its oxidation number to C3 plus. So this is what the question was about. In which environment will there be a reduction phenomenon of C4 plus into C3 plus? And the answer will be acidic medium, because in an acidic medium, maximum number of electrons are donated during the dissociation of H plus from the acid. So what happens in an acidic medium? Acidic medium, there will be H plus ions because of the acid. Let's say HCl is the acid. So when we have an acidic medium, we'll have ions of Cl minus and H plus. So in an acidic medium, maximum number of electrons are donated during the dissociation of H plus from the acid. So there will be free electrons, or electrons are donated. Now these electrons will be taken up by C4 plus, converting it into C3 plus. It is then accepted by the element present in the solution and the element is reduced. So in an acidic medium, an element will get reduced due to accepting electrons. So C4 plus will accept one electron and then be converted into C3 plus by the process of reduction. And these kind of reactions are called redox reaction, in which there is reduction as well as oxidation reactions happening simultaneously. So in an acidic medium, whenever there is dissociation of H plus from the acid, there will be maximum number of electrons getting donated, and these will get absorbed by C4 plus, turning it into C3 plus. So the answer will be, in acidic medium, C4 plus can convert into C3 plus. Now what is a redox reaction? A redox reaction is a type of chemical reaction in which Oxidation states of substrate changes. So earlier we saw the oxidation state of cerium changed from +4 to +3. So that is a redox reaction. So here is a schematic showing the general redox reaction. A plus B with neutral charges. Gain of electron is reduction. B is getting reduced to B minus. So B's oxidation state was 0. Now it is -1. While A loses the electron following oxidation. A's oxidation state was 0. Now the oxidation number increased. A is +1, B is -1. So this whole reaction is called a redox reaction, where there is change in oxidation states of the substrate. Again, I want to reinforce this concept in your mind. Oxidation is always loss of electron or an increase in the oxidation state. When the element will lose an electron, the oxidation state or the oxidation number will increase by +1 or +2. So that process is called oxidation. While reduction is the gain of electrons is always reduction or a decrease in oxidation state, as we see here in element B. So these are the basic two concepts you need to keep in mind while calculating the oxidation number of an element in a compound, or whether to identify which is an oxidation reaction, which is a reduction reaction. So if you have these basics clear, then you can solve complex problems and you can understand what is going on when cerium 4 plus is undergoing conversion into cerium 3 plus. So oxidation is always loss of electrons and reduction is always gain of electrons of an element. Is it clear to the students? Because I saw that you guys had difficulty in solving this question. So I want you to ask any questions if you have any doubt, so that this concept get cleared. Oxidation is always the loss of electrons. Reduction is always the gain of electrons. Is it clear to the students? These are very basic concepts, and your fundamentals should be very strong for you to understand any advanced topics. So I want you to uh, get cleared of these topics. If it's clear, then let's move on to the last question of this session. Question number 10. During mild exercise, the dashed amount of ROS generated dashed. Small amount of ROS generated is required for force generation. Small amount of ROS generated leads to contractile dysfunction. Large amount of ROS generated required for force generation. Large amount of ROS generated leads to contractile dysfunction. Now, which of the following options will be the correct answer? Read the question again. It's a statement based question. I'll give you guys a minute to think about it and post your answers in the chat box. During mild exercise, the dash amount of ROS generated does dashed. Option A, B, C, or D. This is the last question of this session. So any students who haven't gotten a chance to answer these questions, please try to answer. During mild exercise, what do you think is the relation between ROS and muscle activity? Basically, the question is about that. We had many questions regarding ROS and muscle activity because it is an important area of nanoparticle usage. Research of coming up for antioxidant nanoparticle properties to dissolve the reactive oxygen species, which can be used as a supplement for athletics and sports person and people with high muscle activity. Now, which of these following do you think is the correct answer? During mild exercise, what is the amount of ROS generated and what function does it carry out? Come on, students, this is the last question. Just try to guess, even if you are not able to find out the right answer, just make a wild guess. What do you think? Because we have already discussed such questions in the previous slides. We discussed at length about muscles activity, about ROS, about cesium oxide nanoparticles, or glycolysis, and many more questions. So I think you might be able to answer if you have paid attention or you have focused on the earlier slides. What happens during mild exercise? Let me collect some answers, at least 4 5 answers. Neerav has already answered option A. What do other guys think? Please let me know in the chat box. Okay, Roshan has also answered A, and Soumyadeep is also answering A. Let's not waste more time and see. Option A is the correct answer. So Neerav, Soumyadeep, and Roshan, you are all correct. During mild exercise, small amount of ROS is generated, which is required for force generation. That's why doing mild exercise, regular exercise is always recommended, but not strenuous exercise. Only athletes and bodybuilders and other sports person do strenuous exercise, which can lead to high amount of ROS production. Now, this is a schematic or a graph from a research article showing the force versus redox state, uh, redox state of a muscle. So, if you follow this curve, the green area shows reduction reaction, and red area shows oxidation reaction in the muscle. In reduction state, we have high antioxidants and lesser amount of ROS generated, so it is in a reduced state. And in oxidation state, the amount of ROS generated exceeds the antioxidant present in our body. So point 1 here is resting muscle phase, in which we have reduction phase, and isometric force is maximal 50%. And point 2 is optimal force range, in which occurs in between reduction and oxidation state. So mild exercise will lead you to point 1 to 2 optimal state, but doing more exercise or strenuous exercise will increase the high ROS levels, which will again bring down the force, isometric force of the muscle down to 50, and then down even at higher oxidation states down to 0. So that's why a small amount of force is required, small amount of ROS is required for muscle force generation, which occurs in the reduction phase or resting muscle. While in high oxidation states, high level of ROS does not benefit the muscle contraction. It is just detrimental and decreases the isometric force of the muscle. So an optimum level of ROS is also required for maximal force production during muscle contraction. And with that, we have come to the end of our discussion for week 6. I hope it was all clear to you guys. Although this time there were fewer participants, let me just stop sharing my screen. Yeah, again I'm just sharing the YouTube link for recorded sessions of these lectures or practice sessions.
You must have gotten an email from the PMRF, sorry, the NPTEL team about the slides and the recording of these lectures. So I'll be sharing these slides with you as well. But more importantly, I wanted to gain feedback from you guys whether it is clear to you guys, or are you enjoying these sessions? Do you find it informative, or is it helping you in any way? Because that is the main motive of uh, organizing these sessions. So this is just a practice problem session of the real assignments that you are solving here. So you can, yes, Neerav, do you have a question regarding today's session, or you can speak up, or you can just mention in the chat box. I can help you regarding the practice problem sessions that we were discussing, and any other topic related to the course. That's why I'm here, and I hope I need clarification of lectins. Okay, lectins. Wait, let me just switch on my presentation again. I have a perfect presentation for you guys for lectins. In the last lecture series, in the last problem practice series, we were looking at lectins. Let me just give me a minute. Let me share my screen again. I think the question, there was a question on lectin somewhere here in the last practice problem. I hope so. Then I can explain it to you better. I think this is a pectin. Okay. If you would have attended the last lecture, I discussed. Yes, yes. I think this is the question. So lectins are proteins that can bind to carbohydrates. So I hope you can see my presentation. Lectins are basically proteins which can bind to carbohydrates, and they are used in many. They are present on cells variously, doing so many functions. Cellular recognition mostly. Yeah, and they are also used as a pathway for attachment of many bacteria, viruses, and fungi to the cells. So lectins are proteins that can bind to carbohydrates. So if you can, how will I explain you? If you can imagine, let's say a cell has a protein, and the bacterial cell wall is made up of carbohydrates. It's made up of cellulose or peptidoglycan. Peptidoglycan is a polymer of sugar, or peptidoglycan surfaces. Now these lectins can recognize these peptidoglycans, which are carbohydrates present on the bacteria, and the bacteria can lodge into the lectin and attach into the protein, and that's how the bacteria can infect the organism through the lectin. So that is an example of a lectin in uh, which is a protein which can bind to carbohydrates. So these are some of the example of lectins, some scientific examples. And these are lectins are also present in plants which are considered toxic, which are inactivated by proper cooking.
For example, raw kidney beans naturally contain toxic levels of lectin, and adverse effect may include nutrition deficiencies and immune reactions. So they can be allergic. So any protein that can bind to a carbohydrate is basically a lectin. I hope that is what you were looking for. Great, great, Neeraj. Neerav, sorry. Somyadeep, I think he has a question. There was no class for week 4 in the YouTube channel. Oh, I think you should recheck it because till lecture 5 I have all uploaded. So let me just share my screen again and show you guys. I have already shared the link, and if you go through the email provided by you from NPTEL, you can gather the link. Else, I'll just share the link in the chat box again. Let me present my screen again, and I'll show you the YouTube channel. You can just type my name, Ashutosh Behera, NPTEL, or you can just type Nanotechnology in Agriculture, the course name, practice problem set, and you can reach NPTEL. Those recordings of these sessions. See, for people who are not able to attend the practice sessions, that's why there is a recording on the YouTube. You can go and watch there if you need any help or clarification. It's good that you asked. Then, if really there is some problem with the YouTube channel, then I can look into it. Oh, is it? Is it just week 1, 2, week 3, 5? There is no 4. Okay, then I have to really check. So if you see, this is my. So here I have made a playlist, and there are all these lectures. Oh, oh, really, really. Oh, I'm sorry. I'm sorry. Now I see that there is actually no week 4. So you can reach this link here. So I'll just copy this link and paste it in the chat box. Thank you for reminding me. Thank you for reminding me. I'll upload the week 4 lecture, and you guys can all visit this playlist here, and also the email. I'll make sure I'll upload week 4 as well as week 6 lectures. Okay, with that information, let me end this call. Thank you for joining, and see you next Sunday with practice problem set week 7. Bye bye. Take care.